Good afternoon, learners. Today we are going to discuss a very important topic in biology, that is preparation of representative stains, and particularly we will be also focusing on one of the very important techniques, that is gram staining. Now this particular course, this is actually a certificate, part of certificate program in laboratory techniques, CLTL 102, and we would be chiefly dealing with the unit on basic experimentations in biology. So what is the purpose of staining? Basically, if you want to visualize any particular biological organism, then you need to stain it. So that is why this is a very important part which we are going to discuss, that is preparation of representative stains and gram staining. Now, why do we stain? Why do we go for staining? Staining is actually a process by which you can enhance the contrast of any biological specimen at the microscopic level. Now, this staining procedure, it can be of different categories. It can be simple staining, it can be differential staining, it can be gram staining, it can be acid-fast staining, or endospore staining. Now today we will be chiefly discussing with the first three techniques, that is simple staining, differential staining, and gram staining. Now when you need to actually go for any staining preparations, then you have to first, what you have to do is you have to mount the biological specimen. You have to mount the biological specimen on a slide. So you can go for wet mounting, where the living biological specimens are mounted on a glass slide with water and specific stains. You can do it via fixation, that is to preserve the shape of the cells and tissues. Heat fixation can be done to kill and adhere the specimens. Chemical fixation to generate strong bonds and increase the rigidity of the samples. Which are the common chemical fixatives? Formaldehyde, picric acid, methanol, and ethanol. Sometimes you need to couple this staining technique by using a mordant. What are mordants? These are some chemical agents which are used along with the dye to make the specimen stainable, which otherwise is unstainable. So normally they are not stainable, but if you use a mordant, then you can make them stainable. Now, what is actually the chemical nature of stain? Stain is actually, basically they are dyes, and they have two groups, that is chromophoric group which impart color to the dye, and oxochromic groups which render the ability of the dye to attach to the cellular component and to dissolve and dissociate in water, because ultimately you need to dissolve the stain. These are some powders which you need to dissolve. You can dissolve them in water, you can dissolve them in certain other solvents like ethanol, acetone, etc. So the chromophoric group and the oxochromic group. Now these stains or dyes, they can be categorized again based on their chemical nature into acidic dye, basic dye, and neutral dye. Acidic dye, they stain the cytoplasm, particularly the proteins like Congo red, methyl blue, these are some examples. Basic dye, that is the color radical is the cation, they are used to stain the nucleic acids like methyl green, crystal violet, basic fuchsin. Neutral dye, that is acridine orange. As I already said, that sometimes you need to use the mordants because you need to increase the adsorption of the stain to the chemical structure. So in this case, like for example iron or alum, these are some of the very important mordants which you often use. Now coming to some of the representative stains, as I mentioned in my title. First is 1% acetoarsine. Now as I said that you need to stain the chromosomes. You can stain the chromosomes. You can stain any biological organism. As I said, you can stain any bacteria. You can stain a E. coli cell. You can stain a Bacillus subtilis or any Bacillus species. You can stain any plant specimen, like if you cut a section, if you make a histological section of stem or root, then you need to visualize it. You need to visualize the different tissues. Okay, so for all these purposes, what you do is you need to actually stain the specimen. So first we will start with the staining of the chromosomes, because you need to visualize the chromosomes, you need to do a karyotyping. What is karyotyping? You need to count the number of the chromosomes. So in these cases you need to stain it. So in these cases, for the cytological preparations for the chromosome staining, we normally use acetoarsine or acetocarmine. So take a look at how do you prepare this acetoarsine. 1% acetoarsine. Arsine is a basic deep purple colored dye which is obtained from the lichen Rossella tinctoria and Lecanora porella. They are obtained from the parent compound orsinol 3,5-dihydroxytoluene. Now, how do you prepare this arsine? Normally, 1 to 2 gram of arsine powder is dissolved in 100 ml of 45% acetic acid, and then you heat it. Mind that you don't boil it, okay? You heat it up to a simmering point, and then the solution is cooled down to room temperature, and then you filter it. So this is actually in detail the procedure. That is, first you need to weigh the arsine powder, okay? Then you measure the glacial acetic acid, 45 ml of glacial acetic acid, and then you dissolve the arsine powder into it, heat it to the simmering point, then you add water, 55 ml water to it, filter it, and then store it in a bottle or anywhere you can store it, okay? Next we go to acetocarmine. This is another very important stain. Normally 0.5 to 1% acetocarmine is prepared. Now what is the source of this acetocarmine? It is a reddish purple colored dye which is obtained from the female scale insect Coccus cacti, which lives on cactus Opuntia coccinellifera. The bodies of the dried females make cochineal. What you do next? You next isolate the carminic acid. Carminic acid is obtained by extracting cochineal with boiling water, treating it with lead acetate to produce lead carminate, which is again treated with sulfuric acid. The dicarmine is produced by mixing alum with carminic acid. So normally what you do in this case, again you measure 0.5 to 1 gram of carmine powder, and then you dissolve it in 100 ml of 45% acetic acid. You heat it to simmering point, cool it, and then you filter it. So in that way, these are the two basic stains which you use in the cytological preparations. Now, when I talk about the cytological preparation, what is the purpose of making the cytological preparation? Because you need to visualize the chromosome, you need to visualize the morphology of the chromosome, you need to count the number of chromosomes, okay? And this is called karyotyping, and during karyotyping, what you need to do is you need to make a squash preparation. Now, normally in case of plant systems, we use the onion root tips, okay? So this is called squash technique. What is squash? Squash technique is a simple method used for the study of chromosomes. This technique consists of applying gentle pressure on a small piece of stained tissue to spread the chromosomes in the cells. This technique is used to study dividing cells showing either mitosis or meiosis and suitable tissues. Now, one thing has to be remembered, that normally we prefer the metaphase stage in the cytological preparations. Why metaphase? Because you must be knowing that metaphase is the stage where the chromosomes are countable, where the chromosomes, they align themselves at the equatorial region, and so the chromosomes are easily countable. That is why our main purpose is to get as much as metaphase stages in the cytological plate. So for that, what you need to do is first pre-treatment of the root tips. So in metaphase, the chromosomes remain densely crowded together at the equator and are countable. But you have to ensure that all the cells are arrested at the metaphase stage. For that, what you need to do, you need to undergo three steps. First is pre-treatment. What is the purpose of pre-treatment? Disorganization of the spindle apparatus, shortening and flattening of the chromosome arms so as to contract the chromosomes and arrest the cell division. Now here we normally use two kinds of agents. One is colchicine. Colchicine is obtained from the roots of Colchicum autumnale, which is a plant belonging to the Liliaceae family. So normally 0.05% to 0.5% of this colchicine is used, and you need to treat it. Pre-treatment should be done for 2 to 6 hours at room temperature. In addition, you can also use paradichlorobenzene, a saturated solution of paradichlorobenzene, for this pre-treatment as a pre-treatment solution. Here you need to treat for 3 to 4 hours at 10 degrees centigrade. After pre-treatment, what you need to do is go for the fixation. What is the purpose of fixation? Selective preservation of morphological organization and chemical composition of tissues, rendering the cell contents insoluble with the coagulation or precipitation of proteins and inactivation of enzymes, prevent bacterial decomposition. So these are the three main purposes of undergoing fixation process. Here you can use these kind of reagents like acetic acid, ethanol and formaldehyde mixture, alone or in mixture. Acetic ethanol, you can use them in 1:1, 1:2 or 1:3 ratio. Carnoid solution, ethanol, chloroform, acetic acid, 6:3:1 ratio. So you can use any of these agents or in any of the combinations to undergo the fixation process. Now once this fixation is done, you need to go for the actual staining process. Now what you do here, let's say you are given an onion root tip cells. So what you do is, first you have undergone pre-treatment as I discussed, then you have undergone fixation, and then you take this tissue or the onion root tip cells which you have placed in the fixative and you wash it in water until the roots sink, drain off the water and then put few drops of 2 normal hydrochloric acid and leave it for 10 minutes at room temperature, and then you very gently and carefully while the hydrolyzing process is going on, you move the watch glass over the flame, but be careful, don't overheat it. Alternatively, you can just warm the watch glass over a beaker of boiling water till the liquid steams. So mind that the time should be very important in this connection. You should not overheat it. Now after this hydrolyzation is done, you drain off the hydrochloric acid and then what you do is you again wash it with water, and the next step is you transfer it to the staining solution. Mind that you have already prepared the stain, 1% acetoarsine or 1% acetocarmine, whatever you are using. So transfer these tissues to the stain and then stain it for 10 to 15 minutes. So once this is done, then what you do is place a drop of 45% acetic acid on the root tip and carefully place a cover slip. And now the actual squash technique comes into the picture, because now what you have to do is you have to gently tap the cover slip by the back of a pencil to get an even spread of chromosomes. So because you need to evenly spread the chromosomes, they should not clump together because you need to count the number of chromosomes. So you should have applied a uniform pressure to evenly spread the chromosomes and then go for visualization under microscope. So take a look at this picture, how it looks. Like if you stain it with acetoarsine, and this is on the left hand side, you can see it's a typical metaphase plate, okay, where the chromosomes are countable and well deciphered. And on the right hand side, it is acetocarmine, where I have actually tried to introduce you to the different stages, like the anaphase stage where the chromosomes have segregated to the 2 poles. Okay, then there are some stages for the metaphase. So different stages are available in the slide, which is, or in the picture, which is in the right hand side. Okay, the second stain, that is aqueous methylene blue. In this case, if you want to prepare a 1% methylene blue, then weigh 1 gram of methylene blue powder and you dissolve it in 100 ml. It is very simple. Okay, only you have to properly weigh the amount and properly measure the volume of the distilled water. Okay, so in that case you can prepare a methylene blue stain. Now methylene blue is normally used to check the epithelial cells which you obtain from buccal smear. So in this case what you have to do is rinse your mouth well with tap water, gently scrape the inside of your cheek 8 to 10 times with the flat end of toothpick, add a drop of 0.9% sodium chloride or physiological saline on a clean glass slide, then add a drop of methylene blue and then cover it with a cover slip, and again you flatten the cells. And of course you need to drain off the excess stain. You can use tissue paper or paper towel for that, and then place it under the high power of the microscope. So this is how it will look like. If you see it, if you stain it with methylene blue and if you visualize it under microscope, then the cells appear crumpled and irregular in outline. Nucleus, it stains dark blue in the center. And if you have isolated this buccal smear, or if you have isolated the sample from females, then in addition to the above features, you will notice one additional feature, that is bar body. What is bar body? It is a distinctly dark staining body attached to the nuclear membrane. So you can visualize this bar body in case of samples taken from females. Okay, next the staining solution that we are going to prepare is the Noland's stain solution. Now in this case, what you have to take is, you have to take 5 gram of phenol crystals, okay, and then you can measure it and take it in 10 ml of the Corning tube, okay, and then measure 100 ml of distilled water separately, and also measure 10 ml of glycerol in another measuring cylinder, and then 25 mg of gentian violet. You have to measure it, you have to weigh it, and then the phenol crystals which you have already solubilized, or which you have already prepared, okay, by heating it, then you add it to this 25 mg of gentian violet. Also you measure you add the requisite amount of glycerol that you have already measured. And finally you gently pour the prepared stain solution into a brown paper covered bottle or brown stain bottle of 100 ml capacity. So basically it consists of a mixture of phenol crystals, water of course, glycerol and gentian violet. So together they make up this Noland's stain solution. Now where is it normally used? It is normally used for the flagellated algal samples. Now let's take example of Chlamydomonas or Volvox. Okay, these are some green algae which have the flagella, and here the flagella can be visualized using this kind of stain. So place a drop of culture, algal culture maybe, or you can take any flagellated culture on a clean slide, add a drop of 10% methyl cellulose to restrict the movement of the organism, place a covered slip to observe the flagellar movement under the microscope. Stain the specimen with Noland's solution, and as you can see in this particular diagram, okay, that you can clearly visualize the flagella using this particular stain if you stain it with the Noland's solution. Next we come to plant specimens. Now if you are asked to make a tissue section like a stem section or a root section, so in this case what you do is you have to cut the tissue, or you have to make a transverse section of the tissue, okay, and make sure that you don't make oblique sections. And after transverse section you need to again stain it. So in this case the two stains that we normally use are light green and safranin. So light green is a coal tar dye, acidic, and belongs to diamino triphenyl methane group. This is an ideal stain for cytoplasm and cellulose cell wall. That means mostly the parenchymatous tissues. These are visualized by staining with light green. So you prepare light green by dissolving it in 90 to 95% of alcohol. It may be 90% or it may be 95%, or sometimes it may be in absolute alcohol. Normally we use 0.2 to 0.5% solution. Safranin, it is a coal tar dye. It is a basic dye and belongs to the azine group. This is very much specific for the lignified tissues. That means thick wall tissues, lignified, cutinized, suberized and chitinized tissue. I give you an example like xylem. Sclerenchymatous tissues, which are thick wall tissues, they normally take up the safranin stain. So you can get a contrast. If you make a differential staining using these two stains, you can very clearly visualize the tissue sections. Now, how do you go for this staining? So these are some of the steps. Choose an appropriate thin non-oblique handmade section kept in water in a watch glass. Transfer the sections in 30% alcohol for 5 minutes and then go for the staining. First you add 1% safranin. Now this 1% safranin has been prepared in 50% alcohol. Stain it for 30 minutes. Then you go for the series of dehydration steps. Dehydration first in 70% alcohol, then 80% alcohol, then 90% alcohol sequentially. And each of these treatments should be subjected for 5 minutes. Next, you add the counter stain. Now, the counter stain in this case is 1% light green in 95% alcohol. So, you counter stain it with light green for 1 minute. Mind that these time limits need to be adjusted. Okay, I have given a rough estimate of the time, but sometimes depending upon the specimens or depending upon the tissue sections, these times need to be adjusted, or this time need to be modified. Okay, or you need to standardize the time durations. Pass through alcohol xylol 1:1 mixture for 5 minutes, clear in xylol for at least 10 minutes, and then after all this staining procedure is complete, after all these treatments are done, you take it in a slide, put a cover slip and mount it in Canada balsam for making of the permanent slides, and finally you dry it on hot plate for 24 hours. Next day when you come, you will be able to visualize tissue sections like this. See, the red portions are those which have taken the safranin stain, and those are actually the lignified tissues or the thick wall tissues. As I said, it can be xylem, it can be sclerenchyma, and all the light green portions, okay, those are actually they have taken up the light green stain, and those are thin wall tissues, cellulosic masses. Okay, mostly they are parenchymatous cells. So in that way, you can see how beautiful the tissue section look like, and you will be able at once to see each of the layers. Okay, the starting from epidermis, cortex, okay, the endodermis, the pericycle, the pith, xylem, phloem, all the tissues you will be able to make out when you go for this kind of staining. Lastly, I will come to another important technique, that is gram staining, and this is very much important to technique in order to distinguish between a gram positive bacteria and a gram negative bacteria. Now before you go for gram staining, you have to understand the principle of gram staining. Now gram staining technique was introduced by Christian Gram in 1884, and here the fixed bacterial smear is subjected to the following staining reagents like crystal violet, iodine, alcohol, safranin. Now principle is gram positive bacteria, they retain crystal violet color and therefore they appear deep violet. Gram negative bacteria, upon alcohol treatment, they will lose the crystal violet and they will take the counter stain, that is safranin, and hence they will appear red. Now why this difference? Because the cell wall of the gram negative bacteria, they have thinner peptidoglycan layer, but they have in addition they have a lipopolysaccharide molecule, okay, which can be easily solubilized by the alcohol, whereas in case of gram positive bacteria they have a thick walled peptidoglycan layer and therefore that is not solubilized by alcohol, so that when you wash it with alcohol, the crystal violet color, okay, that will be faded away and they will take the counter stain in case of gram negative bacteria. But for gram positive bacteria, they will retain the crystal violet color and hence they will appear violet in color. So CVI complex can be extracted and hence decolorized. So they take safranin counter stain in case of gram negative bacteria. Gram positive bacteria, they have lower lipid content, they have thicker peptidoglycan layer, therefore crystal violet iodine complex cannot be extracted, therefore they will retain the crystal violet color. Okay, so that is actually the steps. So you need to first take the with an inoculating loop, you take the bacterial specimen, you take it on the slide, and then with another glass slide you just make a smear, okay, and then you add a drop of crystal violet, okay. If it is gram positive bacteria, it will retain the crystal violet color, okay. Following crystal violet, you wash it with alcohol, okay, and then you add counter stain, that is safranin. So if it is gram negative bacteria, it will take the color of the safranin. So this is how it will look like. So if you observe your slides under a compound microscope, you can see the gram negative bacteria that has taken the safranin stain and the gram positive bacteria that has retained the color of the crystal violet. So these were some of the techniques that I wish to talk or I wish to share with you today. So you can find these protocols in your lab manual. Okay, please go through it, and in case of any problem, you can approach us with any kind of queries. We are always there to help you. Thank you.
We wear clothes on a daily basis. But have we ever wondered, where do our clothes come from? Who makes our clothing? And who are the people and the process responsible for the whole manufacturing of our clothing?